Okay, I think we should start the session. Can somebody let me know whether my uh, voice is audible and whether my screen is visible or not?
So your screen is visible and audible also.
Okay, that's great. Right, this too. Okay, so should be working just fine, I guess. Yeah, so a very good evening to you all and a very happy Republic Day. So I welcome you all to the live tutorial sessions for the course Bioinformatics Algorithms and Applications. This is an NPTEL course that is instructed by Professor Michael Gomiha from IIT Madras. And alongside him, two TAs will be kind of joining for these live tutorial sessions. And one of them is me, of course. So I am Sunanda Samanta. I'm a PhD scholar at IIT Indore. And basically the kind of purpose of these live tutorial sessions is that we'll go over some of the concepts uh, and some of the things that Professor Gomi has talked about in his lectures, of course. And side by side, we will also talk about certain problems. For example, questions similar to your assignments, what you might be getting in your assignments and your exams. And we'll try to solve those things, solve those problems alongside, discussing some of the concepts, right? So that's kind of the overall gist of it. And I guess you guys have gone through the lecture of Professor Michael Gomia, is that correct? So the week one lecture, so this class is for week one. And in each week, two of us TAs will take the sessions. One is on Monday, mine from 6 to 8, and another, I guess, is on Tuesday, right? Each Tuesdays. So the purpose of kind of having two separate tutorial sessions is that you might not be able to join, for example, a particular one, right? So based on your timing and your convenience and your work and your current schedule, so whichever TA session you feel you're comfortable joining as your convenience, so you might try to join those. If you are unable to join any of those, that's fine, because the recorded sessions for these lectures or tutorials, we will upload it to YouTube later, and we will share the link with the NPTEL team, and they will subsequently share it with you. So you will have the recorded lectures accessible always in this particular YouTube links. Through this particular YouTube links, you will also have the slides accessible, which will be provided via a Google Drive link. Yeah, I see one hand in the chat. Yeah, you can unmute yourself, I guess, and say if you want to ask anything or say anything. You can also type something in the chat. I hope I'll try to see the chat in another screen. So yeah, so I think you guys have gone through the lectures of Professor Michael Gromiha. Could someone brief me a little bit about what Professor has talked about in week one's lecture? Yeah, I see hands in the chat.
Yes,
Yeah.
yes, sir. Can I, sir? And sir, in week one, he talked about introduction of bioinformatics, and then he said that bioinformatics stands for bio and informatics, in which we integrate sciences like computational biology, mathematics, life sciences, and informational physics technology. And with this integration, we can actually deduce long sequences, and we can formulate structure by these sequences. And we have certain databases like GenBank, DTBJ, and all. So he summarized most of the database in his database guide, and last two lecture database and database categories. He talked about DDBJ, GenBank, and EMBL, PubMed databases. Then he also covered the basics of nucleic acid in his week one lectures, in which he talked about the structure of nucleic acids from start to finish. And that's I can remember. That's all I can remember.
Yeah, that's fine. So it was basically a kind of broad introduction to the field of bioinformatics, right? Which again we will also start with. So if you talk about bioinformatics, it is kind of an intersection point between multiple disciplines, right? So firstly, you will have biology, physics, and chemistry. Now, biology is kind of the main thing here, because without the domain knowledge of biology, you will not have bioinformatics. You are basically trying to apply every other disciplines and taking certain principles from different disciplines onto the field of biology to understand biological data. So that is kind of the purpose of bioinformatics. So the main thing here is the domain knowledge of bioinformatics. However, not only that, alongside the knowledge of biology, you will also need the fundamental sciences. For example, in physics and chemistry, right? Those will come in handy while solving different problems in biology. You can try to basically understand biological problems in terms of the basic physics and chemistry. So that's one thing. Uh, Another part is math slash statistics. So of course, solving different biological problems, you will need the fundamental maths as well as fundamental statistics. You will need to verify the significance. You will need to derive different hypotheses. You will try to formulate different algorithms, and math, statistics, all are integrated in all of those. So in just generally in solving problems, you need the help of math slash statistics. Then again, there comes computing as well as information technology, right? So computing and information technology are also necessary because let's say you are trying to again devise different algorithms. You are trying to store large amount of data. You are trying to do raw logic analysis. For that, you need the information technologies. And of course, computing power and the computing resources. These are all necessary for the entire kind of breadth of bioinformatics, if you can think of it in that way. And as I said, the intersection point of all of those is what bioinformatics basically is, right? So it combines the different principles of all of these fields together for solving biological problems, right? So if you just talk about in some terms, so it is basically an interdisciplinary field which bridges computer science, biology for managing biological information, for example, such as DNA, RNA, and protein-based informations. So biology, computer science, information technology, core sciences, all of those are needed. All of those are combined together to analyze the data, and that is kind of overall the broad field of bioinformatics gives us. Now, if we talk about, for example, the complexity of a biological system, as Professor Michael Gromiha also mentioned in his lectures, so throughout the hierarchical, basically complexity of a physiological system, we can have applications of bioinformatics in basically each of those tiny subfields, basically. So, for example, you first have nucleotide sequences. So, our nucleotide sequences uh, is there any importance of this in bioinformatics analysis? Can anybody tell what can you do in nucleotide sequence analysis in bioinformatics basically?
Right. Nucleotide sequences is a part of life sciences, so it provides the data for the analysis.
Yes. So nucleotide sequences, it can provide certain data related to different physiological phenomenon. For example, you can use the sequences for calculating certain sequence-based parameters. It could be some thermodynamic parameters. It could be rigidity, flexibility, right? And it could be various other sort of analysis. For example, you can try to align sequences. You can try to match them, try to find out whether there are different uh, conserved domains that you are getting from different sequences and analyzing them together. So those sort of analysis will give you, let's say, idea about motif discovery, idea about phylogeny, idea about sequence conservation, etc., and so on, right? So nucleotide sequence, definitely a big part of bioinformatics analysis. Then if you go up a notch, a little bit more complex kind of way, right? So you have nucleotide structures, right? So the sequences, the primary sequences, they will kind of fold in a 3D environment to some sort of structures, right? And in the case of structural analysis also, you will find that bioinformatics is a very, very critical part of it. So the structural analysis, for example, you will need to calculate uh, the different interactions between biomacromolecules. It could be like interactions between nucleotides and proteins, or you are trying to block certain regions of a nucleotide using some inhibitors. So for those, all of those kinds of informations, and let's say what certain changes, it could be inside changes, it could be environment changes, what certain changes do to the structure of the nucleotides. Those are kind of the broad area of fundamental studies that you can perform. And for that, you will need the nucleotide structure. So structural analysis is also a big part of bioinformatics as a whole. Then you can also, of course, try to get the gene expression profile. So for example, which kind of genes are expressing under different conditions, how are they expressing, right? these different kinds of expressions you can compare and contrast together. Then if you go up a notch, now from the genetic level or the DNA level, we are moving to the protein level, right? So the protein sequence, uh, of course, the same as nucleotide sequence. You can use bioinformatics to calculate different sequence-based parameters. For example, you can identify through a short stretch of sequences whether these are predominantly hydrophilic or hydrophobic, whether these proteins could reside in, let's say, in a transmembrane part or in a cytosolic part, right? The different parameters related to those and all. So you can try to capture them via protein sequence. And then again, if you go up a little bit in terms of structural, in terms of complexity of the system, so you get protein structure. So the same as nucleotide structure, here you will get the 3D profile of the protein. Of course, as we know that the three-dimensional fold of a biomacromolecule is critical to its function. So the protein structure and function, they are kind of very intimately correlated together. Uh, and you have to analyze those. You can integrate or incorporate different bioinformatics tools. So protein interaction, protein drug interaction, trying to find out any inhibitors, trying to find out whether certain mutations on these proteins, whether they make changes on the structures or not, and subsequently finding out whether they make any functions or whether they have impact on its primary function or not. So these kinds of analysis, the analysis of, for example, protein folding, uh, and protein unfolding and those kinds of pathways. So for that, you can also incorporate bioinformatics in it. Then, as I talked about, a protein interaction. So interaction between different proteins, protein nucleic acid interactions, protein drug interactions, and so on. Ultimately, a big pathway of drug discovery, or for example, designing vaccines and all. So those are also part of bioinformatics, you can say. Then if you move towards a kind of more broader scale of system, you get the cell and the cell signaling parts, right? So cell signaling is basically interaction between different components, right? Different macromolecules. For example, a protein is sort of interacting with its downstream partner, and then that downstream partner is also interacting with something else, and that's how this signaling is proceeding, right? One after another, one macromolecule after another macromolecule, right? So if you understand protein interactions, if you understand the interactions between different proteins, well, I see someone says screen is not visible. So is it visible, or is it, wait, was it visible earlier
Sir,
or not?
sir, it's visible. Yeah, sir, it's
Okay,
visible.
okay, fine. Yeah. So yeah, so cell signaling is basically kind of, if you divide the broad scale into multiple small scale problems, so you'll get basically biomolecular interactions, right? So if you understand biomolecular interactions, you will understand cell signaling. Uh, there is one catch, because if you try to model a entire cell, or like this, for example, a large part of cell signaling, you will need to have very large computing resources. So that's one thing, that's one catch here. So if you try to model, so the same way that you model a protein or a nucleic acid, you cannot use the exact same method for modeling an entire cell, let's say. So you have to approach it kind of differently, but it still can be done. For example, you can use some sort of coarse graining model to build an entire sort of cell and its different functionalities. It will not be the most accurate thing, but to get a broader scale, you will need to sacrifice some of the internal nuance basically, right? So I'm still seeing someone saying that presentation is not visible. Again, can someone confirm? Okay, I'll try to speak slowly also. Yeah.
It's visible, sir.
Okay, I see. There could be some internet issues, connectivity issues in between. So if something is amiss, then please let me know as well, right? Okay, yeah. So that is cell part, cell signaling part, and then if you move again to a little bit more kind of broader scale, you will get the tissues, right? So tissues, organs, and then different physiological models or organisms. So the different studies that involve these, different biological studies that involve this, they also, of course, generate a lot of data, right? So it could be, for example, tissue regenerative medicine or some organ modeling or some physiological phenomenon, right? So those data that have been generated by the experimental groups, definitely you can try to analyze those data using different bioinformatics approaches. And of course, as with the others, you can also try to model the different kind of systems in this way, so through different approaches. And that could be one part of your bioinformatics analysis. This was taken from Sir's lecture 2 slides. So that is the kind of the broad idea of the entire complexity of different biological systems and how you can incorporate bioinformatics in all of those. So just to this was just to let you know that it is possible to incorporate this kind of informatics analysis in every biological system that you can kind of think of, right? Okay. So if we try to talk about the different applications of bioinformatics, um, broadly, these can be classified into, I would say, three different categories, as Sir also kind of discussed. So there is one part structural analysis, there is another part sequence analysis, and then there is another part functional analysis, right? So if we talk about all of those, so in structural analysis, you will, for example, have nucleic acid structure prediction. It could be one thing. So always the structure predictions are kind of very critical in the bioinformatics studies, or you can say broad biological studies also, because as I said earlier, that structures are correlating with functions, right? So if you know what particular structure, the 3D fold or the tertiary fold, multiple domains, etc., that a particular macromolecule has, then you can try to identify their functions as well, because ultimately those folds and cavities that are forming uh, determine a lot of their functions, right? So in structural analysis, you could have nucleic acid structure prediction tools. Then of course there is protein structure prediction as well. It's a big part. Can somebody tell me an example of a protein structure prediction tool that is, for example, recently has generated a lot of attention, and it has kind of improved the field by quite a lot?
AlphaFold?
Yeah, exactly. AlphaFold, right? So we'll talk about AlphaFold as well in some of our later kind of classes. So AlphaFold basically the developed deep neural network model by DeepMind of Google, right? And if you remember, in 2024 chemistry, uh, the CEO of DeepMind, Demis Hassabis, as well as the principal scientist working in the AlphaFold project, John Jumper, they collectively shared one half of the chemistry Nobel Prize. So it's a huge deal in the protein structure prediction community. It's a huge deal for all of the bioinformatics analysts, and let's say wherever we are needing, in whichever studies the structure of a protein is needed, uh, for example, in drug discovery and so on. So in those kind of studies, AlphaFold is a big help. I see. Can you show the previous slide once? Yeah, we can go back a little bit. So in this slide, we just talked about the complexities of biological systems, kind of, and we just tried to kind of show that in every kind of complex system of biological nature, you can implement bioinformatics in one way or another, and it can give you good results. For example, we have sequence-based analysis like sequence-based parameter calculation, thermodynamic parameters, rigidity, flexibility for nucleic acids, as well as for protein sequences. You can have the hydrophobicity, hydrophilicity, and so on. Then you have structure prediction for protein and nucleic acids. Then you have protein-protein interactions, protein-nucleic acid interactions as a part of, let's say, cell signaling, right? And in higher order studies and broader scale studies, for example, tissues, organs related to physiological phenomenon, organism, etc. So those kind of studies will also generate a lot of data, which can be analyzed by bioinformatics. Essentially, that's kind of the gist of it, right? So yeah, so in structure analysis, we have nucleic acid structure prediction. We can have protein structure prediction. We can have protein structure classification as well. So based on the secondary structure of a protein, we can perhaps classify them using let's say in different ways. For example, we can have predominantly alpha helical proteins, predominantly beta sheet rich proteins. We can have alpha by beta, alpha plus beta. These are the different kinds of structural classifications that can happen, or that the proteins can be classified as. So that is also one part of the structural analysis. Then you can have structure comparison, of course. So while comparing different structures, you can try to see which kind of domains are folding similarly. For example, uh, through homology modeling, for example, if you have any unknown structure, you can try to build those. So that is also kind of a part of structure comparison, structure prediction, broadly speaking. So that's the part of structural analysis of proteins. So by comparing the different structures, you can also basically, one example, you can try to see conformational changes under different modifications or different environments. So you can try to get, for example, effects of mutations, uh, effects of any kind of post-translational modifications. So those kind of analysis you can also do on the structural scale. So that is kind of the gist of structure comparison of different proteins, or could be the same protein under different environments or different modifications. Then you have sequence analysis. So sequence analysis, you can do genome comparison. You can do phylogeny. Phylogeny is basically the evolutionary analysis, basically how the different sequences or different genomes are connected throughout the evolutionary kind of time scale. You can get an idea of that through the sequence analysis. Then you can have also gene promoter prediction. So gene promoter prediction is basically, for example, you can try to find out which regions upstream the RNA polymerase will be binding and so on. So these different kinds of analysis you can do. Then you also have motif discovery. So motif motifs are kind of like stretches of sequence, you can say, which are very critical to the protein. And when you analyze the similar classes of proteins across, for example, different organisms, you will find that those sort of stretches of sequences are very similar, right? So you can do, what you can do is you can do sequence alignment, for example, of different uh, proteins that perform basically in the same way, right? Or that does the same function, uh, and then if you align them, there is a chance that you will see that short stretches of sequences are conserved among all of these, right? So why are they conserved? Of course, it could be because that those sequences are needed for the function of the protein. So these short stretches of sequences are what we could call as different motifs, right? So to give you a like an example, for example, uh, there are a group of proteins which are called kinases, right? Kinases are proteins that perform phosphorylation. How do they do that? So there is a cavity. There are, let's say, two cavities in the catalytic domain of a kinase protein. It's an enzyme. So one is an ATP binding cavity, and another is a substrate binding cavity, right? So in the ATP binding cavity, the ATP binds. In the substrate binding cavity, the substrate binds. And what happens is that the gamma phosphate of the ATP gets transferred to a certain accepting amino acid residue of the substrate, right? So this is called phosphorylation, and this induces some sort of structural or conformational changes in the substrate, which could be required for certain functionality, right? So that's the gist of it. Now, if you compare the kinase sequences for different organisms, let's say, um, you will notice that certain parts of the sequences are kind of aligned in most of the cases, right? So one, for example, aligned region could be, if you look at a very functional region of the protein, which is called activation loop. So the initial three sequences of this activation loop is called a DFG motif, right? So D is for aspartate, F is for phenylalanine, and G is for glycine. So what this aspartate kind of does is that, because the ATP kind of binds in a ATP Mg complex as an ATP Mg complex, right, in the kinase domain. And the coordination or the positioning of this Mg, which is a positive charged ion, right? So that requires the negative charge of the aspartic acid of this DFG mode. So this is particularly one short stretch of sequence that is required for the functionality of the kinase, and you will see it throughout kind of all of the kinases that you will analyze, sequence alignment, for example, right? So these kind of different motifs you could try to identify through sequence analysis, which could tell you the functional regions of uh, the particular sequence stretch, right? Or the particular, the entirety of the sequence, for example, if it is a protein sequence. Then you can, so you can also have sequence database searching. So if you have a particular sequence, you are trying to find out some certain matches, certain similar sequences and so on. So you can do database searching based on those sequences. Then you of course have sequence alignment, which is kind of goes hand to hand with many of the things that we talked about earlier: genome comparison, phylogeny, motif discovery, and so on. So sequence alignment is basically you try to align different multiple sequences basically, and you try to find out this could be different ways. For example, one could be global alignment, where you try to maximize the entire overall match between the sequences. So this could be helpful in something like phylogeny or evolutionary analysis. Another would be local alignment. So local alignment will emphasize more on short stretches of sequences, the local particular regions, which could be functional, and that is how the motif discovery kind of works. So different types of sequence alignment will give you different types of results. And then you talk about, if you talk about functional analysis, for example, you could have metabolic pathway modeling. So pathway and the interactions between different metabolites, let's say. You could have gene expression profiling. So how under different environments, the gene expression could be changing, those kinds of analysis. You could have protein interaction prediction. So interaction between different proteins, protein nucleic acid, protein drugs, etc., are very important in drug discovery, vaccine development, and others. And you could also have protein subcellular localization prediction. So where a particular protein is localized in the cell, because a lot of identified proteins, they do not have experimental uh, data regarding the localization, right? So those kinds of analysis based on different parameters, features of the proteins, it could be structural features, it could be sequence features. You can try to understand those proteins or nucleic acids, DNA, etc., in a more detailed way. So that is kind of the overall broad view of the applications of bioinformatics. Okay, just a moment. Yeah.
Sir, can you give me an example of how to find a protein whereabouts in the cell?
So if you're talking about subcellular localization, uh, I do not know any particular tool. So for example, so this could relate in different ways, for example. So of course, to build a particular model where a protein is located, you will need to rely on certain experimental data of other proteins, which you know the localization of, right? Uh, for example, if there is available experimental data on certain proteins, then what you can do is that you can try to build a model that will relate the sequence of that protein, or perhaps the structure of that protein, or something of that sort. And then you can try to feed the algorithm, or you can try to train the algorithm in a way that, given an unknown sequence or an unknown structure or whatever, unknown features basically, it will try to tell you where a particular protein is localized in the cell. So that's kind of the basic idea. uh, I think there are some tools regarding that. I will need to look it up, but that's kind of the overall idea. For example, it could be nucleus, cytoplasm, mitochondria, or whatever, ER, Golgi, body, etc. Okay, so you need to extract certain features from the protein of the already available data where you have the data. You need to relate um, the features with the localization data of the protein, and then with that built model, then you will try to predict the localization of an unknown protein, for example, one that you have just found out, if that makes sense.
Okay, sir. Yes, sir.
Yeah, so any particular tool, maybe I'll look it up later and share with you. Um, for example, I can see there are some protein language models probably are there. So for example, things such as, let's just wait a minute. Yeah, so different protein language models, etc. For example, PSORT, Target P, etc. There are some tools, online available tools also, that you can use in this regard. Okay. Maybe we'll look into some of those later if we have time. Okay, so combining all of these, what we have as structural analysis, what we have as sequence analysis, what we have as function analysis, uh, combining all of these, you can do you can implement in different ways. Of course, you can perform your own analysis, but another part of it is that you can share your sort of developed algorithm, or whatever you have tried to develop, whatever you have tried to curate. you can share those with the others also. So one part of that is software development, right? So software development is basically when you have built a program using different programming languages, for example, C, C++, and so on. It could be Python as well. And you share that software with others so that the others or the others in the same community, the other researchers, so that they can benefit uh, from your kind of developed software in the same way, right? So it will reduce their time of experimentation and other things as well, right? Similarly, there is another classification called database construction and curation. So does anybody know what databases are, why they could be important? Just curious. Anybody? Anybody can let me know.
Sir, can you repeat the question, please?
Yeah. So what are databases basically, and why could they be important?
So databases, I guess, to put it in a way, they are a site that that is used, an algorithm basically that is used for different researchers to put the data if they have find any, and for newcomers to analyze the analyzed data, and to add on to the new data and to find those data, because there are thousands of millions of proteins in our world, and we cannot just relook at each and every one data in a book, as the professor said. So we have to have some kind of databases so that algorithms find it for you, rather than you have to type it and find it in every search for everything in a book, for example. So they are the algorithm.
Yeah, yeah, I see in the chat also, I guess, large amounts of mostly primary, secondary data, sources of curated unknown, yeah, databases of curation of known information. Yeah, that's kind of the overall gist of it. So it's basically a organized collection of data through which the retrieval and analysis is made very convenient, right? So for example, if we talk about it, let's say different research groups might be working on similar problems, right? And through their own experimentations and so on, they will generate a lot of data, right? It could be, let's see, in drug discovery field, it could be different binding affinity data and so on. So the different groups have generated different data, but those are scattered, right? Scattered across the entire community, scattered across different literatures, because of course their own data they will publish and they will work on it. But for you to get, or for a newcomer to get access to that data, they will have to search through all of the literatures, all of the previous findings and so on. Now, it would have been very convenient if there was one particular database that curated all of those data together, and you could just go through that database and find out everything that happened prior to your particular research, and then you will have a very organized collection of all of this data. You will have links to external sources and so on. You will have very high level of annotation, for example. So it could make your research life very convenient, right? Very conveniently in future kind of purposes, right? So that's the purpose of a database building. And of course, in bioinformatics context, also different sort of biological data that are generated through different experimentations, those are always different databases basically collect all of those data in an ordered or organized fashion. So database construction and curation is basically a very important thing that the scientific community does, and in some cases, very high level databases, or highly impactful databases, they get also published in very high impact factor journals as well, right? So if, for example, if we give, of course, Professor Promi has talked about different databases, for example, DTBJ, EMDL, and so on, like gene sequence related analysis related databases. There are other protein structure related databases also. But for example, if we talk about one particular database, let's say, so okay, if we just give, for example, one, oh, okay, it's just service unavailable. That's kind of Unfortunate. Okay, it's okay. So this is one particular database. It's called Binding DB, right? Just an example. So it's a binding affinity related database. Uh, what it is that here, for example, in a lot of literature, you will have binding affinity between different proteins and ligands, small molecular ligands, right? Uh, so binding DB, what it does is that uh, on all of the target proteins that are available and that are being experimented on, it collects the data of the different experimental groups, the binding affinity, the IC50 values, the KD values, KM values, whatever, so on, and then it compiles it here. So here, for example, if you are working on drug development for a particular target protein, you can search the name of the target protein here. For example, if I give just a common keyword, you can be more specific here. But if I just search kinase, hopefully it will open up something. Let's see. Okay, so it is opening up a lot of the hits from my own keyword, right? Kinase. You will see a lot of things here. For example, if I am interested in MAP kinase activated protein kinase 2, then if I click that, what will end up showing is that a lot of small molecular ligands or drugs and their experimental affinity values, IC50, KD, KM, etc., in a list, basically in a ordered kind of fashion. It will have link to the primary literature. It will have information regarding what kind of experimentation, what kind of environment it was kind of worked on, and so on, right? You will have the ability to sort the data using different parameters. For example, you only want the KD values, so you could also do that. And you will have the ability to download the data through some sort of Excel sheet or some format, right? So that it could be handy in your particular analysis, right? So it's not opening up right now. I guess the server is quite slow. But you can imagine that if you have this kind of data very handy and available to you in a curated form, it will save you a lot of headache of going through the different literatures and finding out uh, the different ideas about and the different experimentations that went on earlier, right? You could perhaps also use this sort of curated data in you devising some algorithms, for example, in machine learning or artificial intelligence, right? So if you are trying to build, let's say, a program or a artificial intelligence algorithm that is trying to predict binding affinity, so that could be very useful. This kind of data could be very useful in that because you will get the data for training, right? Training and validation. Uh, so since it is not working, I'll just leave it at that. But the particular importance of database is that it's an organized collection of highly annotated data that saves you a lot of time of going through different literatures. It's very convenient in researchers and in other fields as well, right? Okay, so that was about databases. Then if we just talk about the significance of the studies, I talked about it earlier as well. So it's to comprehend cellular functions at molecular level by examining something like sequence or structural data. Uh, it could be utilizing sequence and structural approaches for very productive functional problem determination and solving the functional problems in biology. It could be genomic and molecular biology research, biotechnology in biomedical fields as well, in forensic DNA analysis, agricultural biotechnology. Uh, Drug discovery is a huge part, protein ligand interaction that could facilitate rapid discovery of different drug candidates, right? Then you will have protein structure for particularly design of molecules and so on. Then molecular phylogenetics, genomics, etc., and bioinformatics or evolutionary healthcare basically. So in healthcare related setups also you can use bioinformatics and the advanced sort of bioinformatics setups under AI and ML and so on. So it's very helpful in all of these sort of cases. Okay. So yeah, we just talked about databases. So these are kind of the few important parts of bioinformatics. So one, we just talked about databases. And there's computational derivation and testing of hypotheses. So if you have certain hypotheses that you would like to, for example, test or you would like to derive some hypothesis, you can do that computationally via different mathematical models like mathematical equations, algorithms, and so on. So before applying it to different fields, you can have basically a computational perspective on that particular problem. So that's where the computational derivation and testing of hypothesis comes on. Then there are web servers. So anybody knows what web servers particularly mean? Any idea? I see the tools run on a server. Yeah, so kind of it. So web servers are very similar to softwares, right? So softwares is that a program developed by someone to perform certain functions, right? That is what it is called a software, right? Now let's say you have devised some sort of program, or you have devised some sort of algorithm. What you can do is you can upload that functionality onto a web and you can make it accessible to the global community basically. So that is basically a web server that will perform certain functions. Yeah, we do not require to utilize our hardware to perform the analysis exactly. So a web server is basically it will perform the analysis onto the host's particular server. So whatever server you are using, it could be again come very handy in a lot of settings. If we talk about certain servers, so can anybody give me an example of a particular web server? Maybe,
Blast.
yeah, that could be one example. So in particularly in case of drug discovery, uh, there are certain web servers which are very useful for predicting something called ADMET. Okay, so anybody knows what ADMET is? Swiss spread I also see, yeah. NCBI blast. Okay, so does anybody know what an ADMET is in the context of drug discovery?
So it's about
Basically, the
absorption,
property of a
digestion,
drug.
metabolism, and excretion. So it talks about how the drug is absorbed, digested, metabolized. So kind of the lipophilic hydrophobicity of the drug and how it is suited to it.
Yeah, yeah,
Kind of.
correct. So ADMET is basically talking about the physiological, physicochemical and pharmacokinetic properties, kind of, you can say, of a particular molecule. Uh, it stands for, of course, absorption, distribution, metabolism, excretion, and toxicity. So the idea particularly is that uh, similarly, like how you need to find out before you proceed for an experimental setup, similarly, like you need to find out if a particular drug molecule is having a good enough affinity or not to a target protein, right? So that's one thing. But alongside that, it's also very critical to find out whether that particular molecule will have any side effects on our body or not, right? So in a physiological environment, whether that particular molecule will be absorbed well enough, whether it will be distributed well enough, whether it will interact with the different sort of enzymes in our body in some way, right? Whether it will have some toxicity or not, what will be its volume distribution, and so on. If we are targeting a neurological disorders, whether that will the molecule will penetrate the blood-brain barrier or not. So those kind of analysis, of course, you need to do that, do those in an experimental setup, so that you get a more better, I guess, more accurate understanding of these parameters. But before even proceeding to the experimental level, what you can do is that you can use some already developed servers and programs to analyze your particular molecule of interest, so that it will give you an estimation or a prediction of how likely these what would be the likely ADMET profile of a particular small molecular ligands, right? So one such or two such examples are Swiss ADMET, also ADMET Lab 3.0, as I'm showing on the screen. So these particular web servers are trained on already available data. So of course, a large number of small molecular ligands already have experimentally determined values of these different parameters, right? So again, similar to how a machine learning model would work. So these servers, they have it already trained model, which will correlate certain features extracted from let's say the structure of the small molecule, and it will correlate that to the different admit parameters, right? It already had a training set of data for that, and now you are able to predict your unknown compound or a new compound using the same algorithm, right? And it will give you pretty good results with pretty good confidence, like the well-known servers like SwissAdmin or Admit like 3.0. If we just, for one example, go to this um, Admit evaluation tab of Admit 2.0, you will see that they already have an example structure here. So if we just click on the example, it will give a smile string of this molecule, right? So you can paste your own molecule smile string here as well. And then if you just submit the job, it will perhaps take a time. In this case, it didn't take that much time, but it will have the properties of the different types of properties of this particular molecule. You see the molecule structure here. uh, from, let's say, the physicochemical properties such as molecular weight to volume, to the number of hydrogen bond acceptors, donors, how many rotatable bonds are there, what kind of rings are there, etc. All right, so pKa, acid base, Beltic point, boiling point, those kind of informations to the different other ones, for example, Lipinski's rule, Pfizer's rule, etc. So those are also important properties or important, let's say, standards that perhaps need to be upheld in new drug discovery projects. Alongside that, you will have the particular absorption, distribution, metabolism, toxicity, and excretion profiles, and the different scores related to those as well, right? For example, in absorption, we will have a Kappa-2 permeability estimate, MDCK permeability estimate, and so on. In distribution, we will have the volume distribution, blood-brain barrier penetration, the plasma protein binding, which will also determine, how much amount of the drug you will have free inside in your plasma. So how much is not binding to particular plasma proteins. We'll have different cytochrome oxidase inhibitor or substrate information, excretion, we have half-life, etc. And finally, of course, toxicity, which is a major part of any kind of drug development or discovery, right? You will have the different toxicity parameters, whether this drug could be toxic under these type of contexts, right? So, for example, in heart blockers, it will have any influence on the on our heart or not, right? Basically, so skin sensitization, carcinogenicity, and so on, hepatotoxicity. So these kinds of information you can get, and the idea is that before even proceeding to the experimental lab, you are trying to predict, and based on these results, you are trying to discard from a large library of molecules, right? So ultimately, you will have a small amount of molecules, small number of molecules that you will actually test in the wet lab. So these kind of servers are likewise very important. As you see, it reduces a lot of our time, since the research group that has developed these servers have made it available for global. So we should be very thankful to them that it is kind of reducing the cost and time of the research involved in our cases also, right? So that's web server. Then comes the part virtual screening and drug discovery, uh, very related to the ADMET profiling that we just talked about. But instead of, let's say, analyzing for the effects of the drugs on a physiological environment, now we are particularly trying to focus on its propensity to bind to a particular drug target. So I see one question in the chat. What does plus minus means? Okay, so the plus minus, these are basically kind of the significance values, you can say. So for example, if it has 2 plus, then that means that it is very high probable chance. If it has 3 plus, then it means that it has even higher probable chance, probably the highest one, right? The minus kind of goes in the same way. You can get the exact meaning of all of these parameters and what they kind of mean in the value or in the plus minus terms also. You can get this in the documentation for the server here. Okay, so you can go to the documentation, you can find out exactly what each parameter means, what its value range should be ideally, and those kind of things, right? So plus minus are basically kind of confidence score in this case. Okay. Yeah. So can somebody tell? Like I said, so now we are focusing more on the particular target of our interest, and we are trying to discover perhaps a molecule against that target, right? So what is the primary, or what is the entire process of drug discovery, and what is virtual screening in that context? Anybody tell?
Sir,
In
virtual
virtual
screening.
Yeah. Please explain.
Sir, in virtual screening, for example, CS kinase. So our main goal is to that we have to see whether this CS kinase is um, can has in for example we can see that expression of CS kinase in colorectal cancer, so it is increased when a person is suffering from a colorectal cancer. So we have the data phase in which we analyze the CS kinase. We have different types of CS kinase a lot in all of the organism, and then we have when we get the particular protein such as CS kinase, we develop feature. We find ligand, for example, so that acts as an inhibitor for CS kinase. That's why inhibiting the expression of CS kinase, that's and further stopping the hindering in the increase of cancer cells in the cancer patients. Then we can use the lab techniques to confirm the inhibitor section, whether this metal or the anything that is inhibiting the expression of CS kinase or any other protein.
Yeah, good explanation. I see a lot of answers in the chat as well. For example, in silico screening is a pre-screening that is done in order to rule out irrelevant candidates, right? So you can focus more on the particular relevant candidates beforehand. You can estimate the outcome of molecules, yeah, certain rules such as lipophilicity, hydrophobicity, Pfizer, etc. So there can be a situation where we cannot do a live experiment, we can do virtual screening. So, okay, so a drug discovery pipeline, how it basically works, right? So the drugs, how they basically work is that they will try to interfere with certain interaction that is going on in a diseased condition, let's say, right? In a pathogenic condition or in internal disease condition and so on. So of course we talked about cell signaling, right? So this is kind of different biomacromolecules, they are interacting with one another. And that's how the signaling is processing forward, right? Proceeding forward. So if one molecule or one protein is interacting with another protein, so that might induce some change in the second protein, and now the second protein might get activated, and that will also interact with something else, and there will be some change in that downstream protein as well, and so on and so forth. So the signaling cascade will proceed, right? Now, since the signaling will require multiple or let's say different proteins to interact together, so of course that means that each protein has a particular cavity where the other partner is binding, right? So one protein's binding cavity will be accommodated by the other kind of interacting partner, right? Now, if that particular interaction is what you are trying to block, or if that is an unintended interaction, or blocking that interaction will help alleviate the symptoms of our disease, or it will help as a therapeutic approach for this disease. So what we can do is, instead of letting the other physiological partner, or in the disease condition, the interaction partner with our target protein, we can just design one let's say molecule, or we can design some vaccines. For example, vaccines is a slightly different case, but we can, let's say, design a small molecule binder of that protein which will occupy the binding cavity where initially it was intended for the interaction partner to bind. Okay, so now since your target protein's binding cavity is occupied by our designed small molecule, so what will happen is that the interaction partner will not be able to bind to this particular target protein, and then the unintended interaction that was ultimately leading to the signaling cascade of the different diseases or pathogenicity, that will not occur. So that is the drug discovery, how drugs kind of work. Now, if you try to design a particular molecule against a particular target protein's binding pocket, so you can have n number of different molecules, right? For example, there are millions and billions of molecules, small molecules that are present in different large scale libraries. Okay, for example, zinc libraries, enamine libraries, and so on. So if you are trying to find out, okay, which particular molecule will be best suited to bind to this target protein's binding pocket, so you cannot just randomly select a molecule and then go for experimentation, right? Because If the experimentation fails, then you will have to start again. And any wet lab experimental and subsequent, in vivo or even going forward with clinical trials and so on, those are very expensive processes. Those are very thorough and time-consuming processes that you do not want to go through again and again and again, right? So earlier, of course, you did not have the computing facilities and so on to perform any large-scale pre-screening before proceeding to the experimental approaches. But now we have those kind of facilities. So that's where virtual screening kind of comes in, right? So you can design, of course, you can do a rational design of your own molecule. You can go forward with that molecule through experimentation, but there is a chance of failing there. It's better to verify your molecule through some sort of screening algorithms, some sort of physics-based computational programs, that whether your drug will ultimately work or not. So the screening part is kind of a parallel part of that. If you have a large library of molecules, for example, as I talked about the billions of libraries or millions of libraries, or even in case of, let's say, if your research is not that large scale, you just want to focus on a certain commercial library that they have available for a certain target protein. So what you can do is that before going forward with any experimentation, you can do a virtual screening. So you will, through some sort of protocols, which we sometimes call, molecular docking. So through that kind of approach, you will screen uh, the structures that are available in that library, and then based on a certain cutoff score, you will select your probable hit candidates. So a docking protocol will basically evaluate the binding free energy, also, kind of a complementary part of the binding affinity between a protein and a small molecule. And let's say your particular compound or a set of compounds is giving you a score higher than a certain threshold that you would like to go for. So in that case, you can only keep those particular small molecules for your subsequent analysis, and you can discard all of the rest. So that will reduce the search space or reduce the experimentation space for the molecules by a lot. It will save you a lot of time. It will save you a lot of resources ultimately in your future experimentations and such, right? And since virtual screenings, you can do, there are different available softwares for these kinds of analysis. For example, for docking and virtual screening, you have specialized software suites such as Schrodinger. You have the freely available ones, for example, Autodoc, Autodoc Vina, and so on. So there are different ways you can get the binding affinity for a large library of molecules, and then you can discard based on a threshold score, and you can discard the unwanted ones, right? So that's how the virtual screening basically works. We'll talk maybe about it later also. But that is also a big part, that since drug discovery is a big part of where bioinformatics is applied, so virtual screening is kind of also a big part of its application, right? And then there is big data analysis, for example, next gen sequencing. So the next gen sequencing processes also, they generate a lot of data, a large amount of data. And to analyze those data as well, you kind of need help of bioinformatics in a lot of cases, right? Okay, so that was kind of the broad coverage of the different topics. I guess we should go forward with just some sample problems. So these kind of problems will be the ones similar to what you will kind of face in your assignments as well as in your exams, right? So I think you will have, right now you have your week 1 assignments visible or accessible to all of you, right? So you need to submit those assignments to the NPTEL, in the NPTEL server for the course. And remember that the score for the assignments is also relevant, because it will ultimately count towards your the entire credit, or the entire, for example, grade that you will be getting, right? Alongside your main exam that will be held at a different date. So these assignments are important, and in this kind of tutorial sessions, we'll try to see very similar questions that might come to your assignments, right? Yeah. I see one hand, please.
Sir, when I saw the my Professor Romia's video, so he talked about DDBJ and all. So the screen on his because it's the video is 7 years old approximately. So
Yeah.
the screen was different. So how to practice on these databases? Because as a second year student, I don't work with a lot of sequence right now. So how do I practice or expect question from the database category?
Okay, so the interface of the particular databases might be a little different, but the different functionalities that will be associated with those will still be there actually, right? So if you just go through the different options of those databases, you can find out different analysis. Maybe we'll try to look into it later also. But the kind of questions that you can expect is basically the kind of questions that you get in your assignments, right? So these will if you just talk about the questions regarding databases, so those will be multiple choice questions regarding certain functionality, for example, certain terms, certain parameters, and so on. Um, and multiple choice questions or integer based questions like numeric answer type questions, those kind of questions will be getting if that is relevant to a certain database. For practicing, I guess I'll just suggest you will just go through the database and try to see what different functions they have added or what different functions are available or not, and try to find out the particular functionality that Professor Gromiha also talked about, whether you are finding those in the modern or current interface or not. Okay, from there you can go on with the analysis. So just try
Okay,
to
sir.
figure out if you're able to find the functions in the current interface. If you are not, for example, we could talk about it later also in some classes. Okay. So I guess we'll just start with some simple sample questions. So these are the kind of questions that you will be getting in the particular assignments as well as in your exams. So yeah, so I'll just show the questions and the different options that could be available here, and then I guess we will. So what you guys have to do is that you guys have to give the answer in the chat, and we'll just see, right? So I see one question: do you need to learn statistics? So statistics, yeah, it's very good that you have some fundamental ideas about statistics, because in a lot of cases you will need to test for different statistical significance. For example, it could be different tests such as t-test, correlation analysis, ANOVA, etc. uh, so in different cases those kind of statistical tests are needed, and it's good that you have a, a basis of the statistical knowledge or the knowledge of probability and so on, right? So it's good that you learn statistics. But in this particular case, if you're talking about the exams and the exam questions, so the concepts that particularly Professor Gomiha is going through, please try to focus on those main. Okay. Uh, the different kind of examples that are coming in your assignments, try to focus on those and see like what kind of tools you need to understand those sort of problems, right? Because I don't think anything apart from what is being discussed in the course by professor and what kind of questions you are getting in the assignments will come in your main exam as well, right? So we'll just talk about it a little bit, the questions. I see one comment. You are new to it. You have bio background with some Python knowledge. So how should you proceed? Yeah. I see. You see bioinformatics. If you want to proceed with it in some higher studies also, or if you want to try to understand different applications. So whatever knowledge you have, you just try to understand how you can particularly implement that knowledge or that skill set in different applications, right? For example, you're talking about Python. Of course, in a lot of informatics analysis and bioinformatics as well, Python is quite handy language to know because it is basically primarily kind of used for data analysis, right? Statistical models, AI, ML, data analysis, and so on, right? So you can try to see what different tools or packages are available within Python that will help you with certain bioinformatics analysis. For example, it could be sequence-based analysis, structure analysis, and so on. And then you basically try to see whether you can apply your own Python skills or knowledge for this kind of different analysis, or otherwise you can, if you can try to develop different packages also. So that's another thing. But if you have certain biological problems, you just try to uh, guess or try to comprehend how you can use your skill set to solve that problem. So there will be a lot of similar problems and similar softwares that are taking care of those problems already available. So you should search for them, and you can think of it how you can analyze, how you can use your own knowledge in those, and how you can perhaps improve those sort of concepts, those sort of functions, right? So the questions are different than what, yeah, so the questions, some questions are different than what sir taught. So I believe that the questions in the assignment should be rooted in one way or another to what sir has taught. But for example, what kind of questions you are talking about that is not taught by sir? So it should be rooted in one way or another, right? And if there are certain, for example, connections or certain connections that you cannot make with the topic, you can also try to, for example, analyze those questions on your own, and then when the results get revealed, then you can try to compare your answer with the results and so on. Yeah, one question, one hand in the chat also. Yeah, please go ahead. You are pushing BTech in biotech on Dr. APJ Abdul Kalam Technical University. So, what? So yeah. So see, I guess a lot of the concepts that sir will talk about will align with your course curriculum as well. Uh, if some concepts that do not have you do not have in your course curriculum, then just learn those alongside whatever you have on your course, right? So it's good that you have in your course curriculum, you will also pick the bioinformatics topics will also get covered. Then you are taking some additional approaches such as the NPTEL course, where you will learn some additional things as well, perhaps a little bit more application based, or you'll get more idea regarding the applications and so on, right? So it's always good that you have multiple parallel, ways of learning or ways of getting information. BSMS Interdisciplinary Sciences. I'd like to pursue PhD in bioinformatics directly after 4 years of BS. Is it possible? Which university has this course? So, if you are already doing a BSMS in some topics related to biology, you are saying that after 4 years of BS. So, I don't know, BSMS, the entire course curriculum, if it's completed in this 4 years. So, after that, you can just apply for the different PhD programs for different universities, right? So you can apply for example, IITs. You can apply for the state or central universities, the ISARs, the NITs, and so on. All of them have PhD programs available. And basically, what you need to do is that you need to, before joining the PhD, you need to sort out, I would say, a list of uh, topics or research topics that you are particularly interested in. You need to find out which faculties from which universities or IITs or ISARs are working on those sort of problems, because in a lot of cases, when you join under a particular faculty, uh, the research that is going on or the specialty of that particular faculty, so you will get a problem statement perhaps very related or very similar to the research that is going on in the lab, right? So it's best that you sort out in which area you want to do your or you pursue your research on. It could be, for example, just broadly, it could be any computational field, it could be virology, it could be immunology, right? It could be tissue regeneration, it could be nanomedicine, and so on. The different types of uh, topics are there for different faculties. They specialize in different parts. So rule out that, sort out what you want to work on, and then apply under different faculties. Of course, it's very possible. Another, let's say, suggestions I would like to give is that try to crack some of the exams that will provide fellowship for your PhDs, right? Those are, for example, CSI, NET, UGC NET, and so on. In case of biology, you have a lot of other options like DBT, Department of Biotechnology. They have a fellowship GRF program, right? Then TST, if you are a university topper, then you could apply for TST related funds for your JRF as well, JRF, SRF, and so on. So these are different things, and IITs, they also take students, or they give fellowships based on GATE exam as well, right? Because it is technically it is a technological institute, so they also accept GATE related admissions. So try to crack those exams and then try to see what research area you're interested in and try to apply under those faculties and give interviews, take exams. That will help. Okay. What are the tools we need to familiarize with? So the tools that you need to familiarize for this course are basically the tools that Professor Gomiha will talk about, right? So the different bioinformatics tools, it could be sequence related, sequence analysis related tools, it could be structure related tools, it could be some docking tools, etc. So those kind of tools you will be needing to familiarize in this course. Whatever Professor Gomiha is particularly talking about, focus on those because those will ultimately be good for the exams that you will take. Okay, so similar concepts will be there. Yeah, so that's one thing. If you're interested in implementation of clinical trials, data drug discovery, what will be more relevant, R, Python, or some other systems? So it dependent on which language you are compatible in. So I would say a lot of programs, a lot of analysis, statistical analysis you can do in R. So Python is kind of a always expanding project, right? So new libraries are constantly being added in with different functionalities, right? You can develop your own library in Python as well. I would say Python is probably kind of in a modern setting the most accessible and the most kind of well developed and the largest community having language. So I say learn Python, learn R also alongside that. It's a very good one. Is it dependent on whichever language you are? Definitely you can do analysis in a lot of different languages, but the languages that have a big community and that have already a big set of tools established, well-established tools, those will be helpful. So I'd say R, Python, both are good. Python would probably be a favorite here, kind of, if I say like in a broad scale. So that is good. So does all branches of biology like MSc and food tech and work in bioinformatics? So bioinformatics can be applied, as I said, in any field of biology that you can think of. Bioinformatics should be applicable in one way or another, right? Because it relates to development of different softwares, algorithms. It relates to the analysis of biological data, right? So anywhere you can get biological data, it's good for application of bioinformatics. So this course will be useful for anybody in the broad field of biology because a lot of fundamental sort of application of bioinformatics will be for different areas in the entire kind of life science field, right? So it's very useful for a lot of you. And if you want to later pursue research in this particular, for example, computational or bioinformatics field as well, that will also be good for you.
Sir, I have a question.
Yeah, please.
Sir, are there any possibilities students get involved in machine learning and Python internship programs for the students who are already enrolled in this course, whom we should be communicating for that?
So you are talking about whether this course will provide hands-on on machine learning particularly. Is that correct?
No. For interested candidates, those who are specifically requesting for that, is there any possibility we can commit with an internship program while attending the course?
Yeah. So if you are talking, so internship you are talking about, right?
Yeah, that's right.
Yeah. So if I remember correctly, the year previous to the last one, so that cycle's course for this bioinformatics algorithms and applications, they had given, I guess, one proposal where the top students of that course, they will be able to proceed forward with some internship with Professor Michael Gromiha, the course instructor, the principal, basically coordinator of this course, right? I do not know particularly whether they still have this proposal or not for this year's cycle. So what you can do is you can ask the forum directly that if I am interested more on this topic, for example, I am particularly interested in the application of machine learning, AI, ML, etc. in bioinformatics. So can I have an internship? So that's one way. If it is officially given, this kind of internship proposals are officially given via this course. But another way, I would say that you should directly just go for trying to get any internships through the official email addresses of different PIs, right? For example, Professor Bromiha, they have their particular institute email ID. You can get the information regarding other faculties that are working on the same field, for example, in AI, ML, or in drug discovery, or in related fields. So the best approach, I would say, is that you try to contact them via an official email that, see, I am a student from here, I have this skill set in AI, ML, etc. I am more interested in bioinformatics or computational biology and whatever research you are doing. So I'd like to get an internship opportunity at your institute. Okay. So reach out to the supervisors directly via email. That is what I would suggest here.
Okay, sir. Thank you.
Yeah. So as a student myself, I cannot tell you whether the supervisors will accept requests for internship or not. For that, I would say you just go with directly contact the PIs. Yeah.
You have to directly email them, right?
Yeah. Yeah.
Okay. Got it, sir. Thank you very much.
Yeah. So I see other hands also in the chat. Okay. So bioinformatics at Central University Haryana.
So may I?
Yeah. Please.
Sir, I'm currently pursuing my master's in microbiology, but lately I have grown interest into this bioinformatics. And sir, I want to ask you that can you tell me that how will I teach myself bioinformatics starting from the scratch, like to develop those equivalent skills which has a student who is pursuing MSc in bioinformatics. Like I don't know any Python language, any R language, and I don't know from where to learn those languages. So
So
you can suggest them.
So I would say that first get an overall idea about which applications and which fields bioinformatics will be used for, right? So if you have a clear idea on that, then you try to, for example, focus on a particular set of problems here. Okay. So in this course, for example, you will be introduced to a lot of different basis of problems where bioinformatics could be useful, and then you on your own try to see, okay, is there any way that I could, for example, solve this kind of problem using bioinformatics tools or not? So there are different numbers of online resources where you will get this sort of idea regarding the analysis of the data in bioinformatics. Some books are there also, some very old books. For example, I'm also taking reference for a lot of my slides from different books. So if you have an idea, you can go through the particular concepts of the particular application that you are trying to do on those different books as well. So first get a overall idea about what it basically is, and then try to see which tools are there, which tools could be applicable here, and then try to just learn those tools on your own. That's what I could say. Regarding Python or R, so there are a lot of freely available resources on the internet that will basically allow you to learn those sort of languages. So for Python particularly, you will see hundreds of videos on YouTube. There will be, like, for example, 6 to 8 hours, 10 hours, 20 hours long, freely available videos on YouTube where they will teach you Python from scratch, essentially, right? So they will start from the data structures, the different data types of Pythons, and they will gradually move towards more complex kind of topics, for example, different statements, for loops, and so on, the functions, the classes, object-oriented programming, and so on. So they will teach you step by step from scratch. So there is a lot of freely available resources on the internet. I would say learn Python through them. Similarly, for R, you will also find different resources, different forums where they have useful information. So it's just about, I would say, searching, and then you will get the results. So the different resources are already available, I would say. Don't focus if you are already doing masters in microbiology. So that's your main focus, kind of, right? Don't focus on all of the problems of bioinformatics at once. Try to select a particular area of problems that you are mostly interested in. For example, let's say, if you are mostly interested in databases, right? And how to write databases, how to basically create databases, and so on. For that, you might you will need to learn something called SQL, right? MySQL and such kind of languages, right? So those are particularly useful for the database creation, organization, and so on. So if you're not interested in databases, then you do not need to learn that language, that particular language, right? If you are, for example, more interested in something like drug discovery, so that's a different set of field, right? So there you will need to learn about molecular docking, let's say molecular dynamics simulation, admit profiling, and so on. And in that particular cases, you will see different tools are available. For example, as I said, freely available like Autodoc Vina. You might need to learn like protein visualization programs like you say Chimera, PyMol, and so on. So, and then if you are later interested in something advanced like molecular dynamics and so on, so they have different packages. For example, the freely available one is GrowMax. So if you are interested in drug discovery field, so that's the set of tools that you need to learn. So that's separate from databases, right? And then if there is, for example, something else, if you want to analyze, for example, large sets of data like next-gen sequences and so on. So for that, again, you need to perhaps learn some different languages, for example, Python and so on, which have the capability to analyze a large set of data, right? If you are interested in particularly developing programs like servers and so on, for that, again, you might need to learn something else, right? If you are interested in particularly software design or software development, then you might need to learn low-level languages, for example, C++. So just try to identify the particular area that you are mostly interested in, and just search from or try your the search based from there. So a lot of freely available resources are already there. So I don't think you will have a lot of problem basically. And as I said that I will share these slides in a particular Google Drive folder. So alongside that, I will also include some books that I took references from. But these are old books, of course, but a lot of the concepts that are being talked about are very similar, right? So the fundamentals, they don't change. Of course, advances are there. But through by going through those books as well, you might get some idea about the different applications or the different research bases, different fields or broad applications that you can have also. Okay, so I'd say try to focus on small things and then gradually grow bigger in that way.
Okay, sir. And sir, one more thing. The Python language of computer science engineering and Python language of bioinformatics, are they both same?
So see, the fundamentals of Pythons are same across all of the fields and applications, right? So if you are trying to implement Python in bioinformatics or statistical analysis or for even development, right? The fundamentals are same. You still need to learn the basic data types. You still need to learn the loops. You still need to learn the functions and the classes and so on, the object-oriented programming and so on, right? The fundamental will stay same. The difference will be that in the different set of analysis, there are already established packages. Okay. So for example, in software development, you might have a different package. Packages are basically a set of tools that are already developed by someone and they are made available to you, right? So those, instead of writing the code from scratch, from ground up, they will already have large implementation and you can use those packages to perform certain functions, right? Uh, so for software development, there will be different packages. For bioinformatics analysis, there will be different packages. For example, in case of let's say statistical analysis, right? You might need the Python libraries such as scikit-learn, right, where you have implementation of the different data analysis and AI/ML and so on, right? So first you focus on the fundamentals, and then as you learn the fundamentals, then you come to your specific area of interest and whatever Python libraries or packages are available for that particular functionality, then you focus on that. Okay. So first the fundamentals and then learn the specific libraries that will be useful to you. If that made sense. I would say for bioinformatics analysis, firstly, you could try to see, try to go through some course where they particularly talk about data science and statistical analysis using Python. Okay, so there you will have, you know, the NumPys, the scikit-learn, the pandas, and those sort of libraries. So that will be also useful in bioinformatics because it is essentially a data analysis tool, right? Data analysis kind of field, right? yeah, Python for data science. These kind of courses you can also do. Drug discovery, I see. See, there are a lot of resources already available. There are different hundreds of thousands of YouTube videos where you can learn Python and other programming languages. Freely available resources. I think there are some courses on NPTEL also that are given by or provided by the professors at IITs for learning Pythons and other programming languages. So you can search for those also. Is Japan an option for this? Sure. Any country that you want to pursue your higher research in will be fine. So I don't know if you are talking about masters or PhD or something like that, but just look into the different institutes, the foreign institutes that kind of align with your interest, the topics or the courses of your interest, or the particular research field of your interest, and then you can proceed forward with that. That's no problem. Okay. Yeah. So in this question, it was DNA and RNA, the difference, right? So of course, yeah, it's both A and B, right? So there is sugar level and base level differences also, right? So the base level, we know that DNA is adenine, thymine, guanine, cytosine, and then for RNA it is adenine, uracil, guanine, cytosine. For sugar we have deoxyribose for DNA, and then for RNA we have ribose. So these kind of simple questions will also you will also find in your particular assignments. You'll also encounter those. Okay. So if you talk about, for example, related question, so what is the linkage that is present between nitrogen bases and the pentose sugar in DNA? So try to answer this in the chat, and then we'll look at the answer. Okay. So another thing I will also try to include some, let's say, Python learning books that I have soft copies for. Maybe in the folder that will be shared to you. So you might also try to look into those as well. Okay. And later, let's see if I find some interesting research papers that I talk about in this in slides. So I'll also try to include those papers, their PDF files in the folders. Is it certificate acceptable in Australia for further education? So I do not know the particulars about how Australia for their higher education accepts certificate. You will have to find that out through the particular institute's page or the particular education agency's page and so on. So one thing I will tell you definitely that these sort of course, the certificate that you will get from this course as well. So it will enrich your CV ultimately, right? So it will enrich your CV, your resume, and then ultimately that will help you in some ways, perhaps not directly, but as a secondary kind of display of your skill set. This will help you in a lot of sense. For example, whether you try to get some higher education or something like that. Okay, in the job market, maybe right. So not perhaps directly, but definitely via the enrichment of your skill set and your CV, this course will help you. What is the practical application? As I said, right, this course is kind of meant to introduce you to the broad field of bioinformatics, right? And the different applications that it will have. So with that knowledge, now you can try to see the different problems in the biological field, and you can try to apply your informatics knowledge on those, basically, right? So further, if you are interested in pharmaceuticals, drug discovery, you can go in that direction. If you are interested in software development, you can go in that direction, so on and so forth, right? The different sort of applications it will have, different applications. Okay. But I want to study bioinformatics. How should I proceed? The same thing. If you have not studied any computer science or any programming languages, just start right now. It will not be that big of a problem. I'll tell you. So the particular fundamentals of programming languages or Python particularly, let's say, they are kind of, if you start kind of using those languages regularly, it will just become a habit. It's not a matter of whether you have studied earlier or not. And like I said, you do not need to know everything regarding certain programming language. You do not need to know everything. Get the fundamentals first, and then just focus on the libraries or the particular tool set that you will use for your biomedical science analysis or your bioinformatics analysis, and so on, right? So it's not that big of a problem. Yeah, so okay, we'll talk about the question. So this is a glycosidic bond that is present between the bases and the pentose sugars, right? So if you see the sort of structures here, let me get the pointer here. So of course, here you have the sugar, right? It is a deoxyribose sugar because on the 2' position, you will see there is no hydroxyl. So that is a deoxyribose sugar. On the 3' you have a hydroxyl. On the 5' carbon, you have phosphate group attached to this, right? So how the chain formation kind of works is that it works via something called a phosphodiester bond, right? So the phosphodiester bond is that the 5' phosphate group will get attached to a 3' hydroxyl group of the preceding sort of sugar, the preceding nucleotide sugar, and then it will form a phosphodiester bond. But that's not the bond that we talked about in the question. So in the question, it had which bond was used for basically is present in connectivity between the sugar and the base, right? So that is called a glycosidic bond, this particular sort of bond that is present here. So it's a glycosidic bond. That is the phosphodiester bond connecting different nucleotides. And then which bond connects two amino acids? Anyone? In a protein?
Peptide bond.
Peptide bond.
Yeah, peptide bond, right? So it is a peptide bond, for example, one amino acid that has a carboxyl group, another amino acid that has an amine group, and then these two are connected by this CONH peptide bond, okay? And then you also have hydrogen bonds. For example, in guanine and cytosine, you have 3 hydrogen bonds. If there is adenine and thymine, they will have 2 hydrogen bonds. So the hydrogen bonds are basically a difference in partial charge distribution between the attached electronegative atom and a hydrogen. So that creates kind of a you could say delta plus charge on the hydrogen, which is later attracted to some other electronegative atom or group, right? So that is the basis of forming hydrogen bond. Okay. Yeah, so these books that I am referring to here, as I said in the references, I will try to include those in the particular folders, so you can get also access to those books as well. Okay. So anybody can tell what is the complementary strand of 5' to 3' ATCGA? Yeah, on hand in the chat, please.
So I think the folders will be shared through email, right? That we can access via email, right?
Please say that again. Because the voice is very.
Sir, the folders that got the content relevant to this course will be shared through emails, right?
Yeah. So I believe it will be shared through
Or
emails.
do we have to like check it on the website anywhere?
Should be both. So on the website also it should have the link there, and on the particular emails also you'll get the link, I think. So the link will basically be the link to a spreadsheet. Okay. So in this particular spreadsheet, we'll have links to the uploaded videos to YouTube, as well as we'll have links to the slides kind of folder on Google Drive. Okay. So the spreadsheet link you will get through email. It should also be available on the website. And when each course, when each week's course is completed, or each week's kind of tutorial session is completed, we'll upload the videos and we'll upload the slides and so on. We'll paste the links on the same spreadsheet basically. So the spreadsheet will keep getting updated, kind of.
Sir, books also will be available on Google Drive, yes?
Yeah. The slides will be available on Google Drive. I'll try to share the books also, also the ones that I'm referencing. The link to the videos will be available via YouTube. Okay.
Sir, for the main examination, will we have this MCQ type of questions or descriptive as well, or something like
So
that?
I don't think there will be descriptive type of questions. It should be the same as your assignments, MCQ and then numeric answer type questions. So it should be the same in the final examination.
Okay, and these two sessions.
Yeah.
And these two sessions, which are to be held for the next 12 weeks, like on Monday and Tuesday. Will this both sessions be same for means respective weeks?
Yeah. So basically, for one particular week, the two TAs that will cover under the same topics. Okay. The purpose of having two different sessions or multiple different sessions is that you might not have time to join as per your convenience, let's say one session, right? So that is why there are multiple TAs that will kind of cover the same topics, maybe with slightly different approach, but they'll cover the same topics. So the sessions will be similar. And if you cannot join, for example, either of those, then the links will be available on YouTube, the recorded lecture links.
So this the questions and the problems that will be solved will be same for both the sessions.
No. That's not given. So because we both are of course taking references for these questions through the earlier iteration of the course that had occurred in the past years or past few years. But it's not given that we will include the same questions in our slides particularly. Okay. So the questions, because we are differently preparing them, so it could be different.
Okay, sir.
Yeah. So for just a moment to to get the complementarity. So the answer would have been B in this case, right? And as you know how to calculate complementarity, let's just try to do that. A, T, C, G, K, A. If you have 3 times, so what you will do is that you will just kind of add the complementary part here. For example, adenine will have thymine, right? T, it will be G, C, T, T, and 5 prime. And since the question is particularly asking in the direction of 5 prime to 3 prime, so you will have to give in this direction. So it will be 5 prime T, T, C, G, A, T. So that this will be the answer here. Okay. So these types of questions is what you will encounter in the exams. Okay. Let's say a certain type of analytic kind of question. So if you have a DNA binding protein or a set of DNA binding proteins, so the interface residues, so interface meaning the particular interface region where the DNA is binding with the protein. Okay. So the interface amino acid residues will have preference for what kind of particular types of amino acids. So anybody want to give the answer here? Yeah. So as I see in the chat, a lot of you are saying positively charged amino acids, right? So because DNA we know is negatively charged because of the phosphate group, so the binding partner of DNA, the protein, in the interface region, it will try to get or try to keep a favor for the positively charged amino acids, right? So that means that in the interface regions, we will have preference for positively charged amino acids, right? Like arginine, like lysine, like histidine, and so on. Just okay. Yeah. For example, another question we have here. So, which of these databases can give structural information on proteins? Any idea? Yeah, I see a lot of B in the chat. So GenBank, DDBJ, EMBL, these are all sequence kind of based databases, right? And RCSP PDB is the particular database where we have the structural information regarding proteins. If we can just open that up, maybe. Okay. So this is kind of the interface of the RCSP protein, RCSP PDB database. Here, basically what happens is that a lot of groups, they will experimentally solve the structures of different proteins. It also has nucleic acid structures, I guess, for some. and then they will deposit the particular PDB files or PDB structures in this particular repository or in this particular database. So that is the RCSP PDB database. For example, if we just type out, for example, sprint tyrosine kinase, it will show you. I don't know why only one is showing here, but okay, let's again try common search. Yeah, let's. I just typed in kinases, and you will see that a lot of different structures are available here. Anybody knows which experiments are performed to get this sort of structures?
X-ray crystallography.
Yeah, X-ray crystallography. Another one is cryo-electron microscopy that also gives coordinate files. So here you will see that if you just randomly go to the any ID, so these are the four letter accession codes or the PDB IDs as we call it. So if you type in this one in the search bar as well, you will get the same result. You will get the information regarding what protein it is, whether there is a small molecular ligand bound to it or not, the expression system, organism, what type of class of protein it is, class of enzyme and so on, mutations, whether it is available or not. You will also get something called the resolution of the structure. So any idea what resolution is? For example, if I have a structure with a resolution for a protein of 1.1 angstrom and another one is 2.3 angstrom for the same protein. So which one is a better structure? Kind of. ने बताया बिल्कुल नेचुरल और 5 स्टार 2.31 Okay, so in this particular case the resolution will mean that it is how well it is kind of able to differentiate between closely situated points, right? So that's what kind of the definition of resolution is. In the case of PDB structures also it's kind of the same. So how it is able to differentiate the electron clouds and so on. So the lower the value here is, the better resolution structure it will have. So if something has, for example, for the same protein, if something has 1.1, if a deposited structure has 1.1 angstrom resolution, so that is a better structure qualitatively from this particular one, right? But it of course depends on other factors as well. For example, in one particular structure, you might have a multimer structure, right? The different monomers are put together and they have solved the entire structure. So that might have a resolution value a bit higher, so little lower quality resolution. But then you cannot get this particular multimer, let's say, in other deposited structures as well. So you need to consider particularly which one type of study you are interested in and choose your structure accordingly. Okay, you will have the associated literature as well, and any available small molecule that is attached to it, possibly you will get it through here. For example, here is on ADP also, right? So that is a co-crystallized molecule. Okay. So yeah, so that was RCSP PDB. And then can anybody tell how many codons can be formed via AUGC? So I see one comment. Can you get the session in recordings? Yes, the recording is and will be available through you via a shared spreadsheet from the NPTEL team. Where after each session, for example, we'll take some time to upload the video to YouTube, and then we'll get the link of that video and we'll paste it on that particular spreadsheet. Okay, so for all of the sessions that will happen, you will get the associated recording as well. So yeah, I see 64 answer. So that should be the answer, right? So you can think of it in this way. So since codons are formed by 3 bases, so you can think of it like 3 boxes are there, and how many ways you can fill up each box. For example, this box can be filled up 4 ways. This box also can be filled up 4 ways. This box can also be filled up 4 ways. So ultimately it will be 4, 4, 4. And now to get all of the combinations, you have to just multiply, right? So that will give you the answer 64. Okay. So in total of 64 ways, you will get the combination for forming codons. Okay. So again, a similar question: which of these codons will not code for an amino acid? Anyone? Yeah, I see answer that C is the stop codon. Okay. So here you can see that UA, UAG, UGA, these are stop codons. Although in certain cases, as you can see in the footnote here, the UGA pen is not working here. But anyway, the UGA is sometimes coding for selenocysteine as well as UAG is sometimes coding for pyrrolysine also. Okay, so you need to also keep that in mind that in certain, so it's specified here that in all domains of life, so bacteria, archaea, eukarya, so in all of these cases, these sort of particular amino acids that are not standard, of course, but these are found. Okay. So again, the information is taken from the iGenetics book. Okay, this is kind of a calculation based problems and you can get similar problems in the assignment also. So could you try to calculate the composition of positively charged amino acid in the given sequence? So it's kind of a long, slightly long stage of sequence and then you have to calculate the composition of positively charged amino acid. So try to calculate it and then share in the chat. Okay? Okay. Yeah, some answers. I will also try to say in the chat. Just look at it. Uh, yeah, it's fine. It's fine. We'll just show how to calculate. Okay. So what the question is asking, I see a lot of you have also answered C. So it's asking the composition of positively charged amino acids. Now, what are the positively charged amino acids? Of course, you know that those are your lysines, your arginines, or histidines, right? So there is no H here that I can see. But if I just color it differently, we'll see there are 1, 2, 3, 4, 5, 6, 7 total positively charged amino acid, right? So it's just a percentage calculation. Out of 20, you have 7 positively charged amino acids. So you do 7 divided by 20 and you multiply the whole thing by 100 to get ultimately 35, the value of 35. So that is the exactly what the question is asking for. So the composition for positively charged amino acid in the given sequence will be 35%. Okay. So if you are given any sort of question, so just calculate the length of the entire sequence, calculate how many specified type of amino acid is present in this particular sequence. In this case, positively charged. It could have been negatively charged also, right? And then you just divide the particular specified question by the entire length multiplied by 100. You will get the percentage composition. So in this case, it would be 35%. Okay. Yeah. So let's say one question we have. So next gen sequencing analysis. Do you know what sort of sequences they require? What sort of files or parameters they are used for or they are needed? Yeah, so a lot of you are answering B, right? So DNA sequences have been analyzed by the next gen sequencing analysis, right? So does anybody know before the development of next gen sequencing or even currently, like what is one of the standard way of sequencing DNA? Anybody knows the name of the method, perhaps?
So, Sanger sequencing method by Frederick Sanger.
Yeah, Sanger sequencing method is a traditional approach, right? So in Sanger sequencing method, what kind of happens is that, so you have your sequence, there's a pointer, yeah. So you have your sequence. Now let's say you are pouring your reaction mixture into 4 different tubes, right? And in each tube you will carry out a polymerization reaction. But what happens is that on each tube you specifically add a very small amount of dideoxynucleotide. So in this case it was added DDATP, then DDCTP, DDTTP, DDGTP, right? So different types of dideoxynucleotides. Now what are dideoxynucleotides? Of course, the dideoxynucleotides are nucleotides which have their 3' hydroxyl removed. So if you remember that the 3' hydroxyl that is required for forming the chain with the 5' phosphate of another nucleotide, right? So if you have a dideoxynucleotide and it gets incorporated during the polymerization, what will happen is that it cannot further take an amino acid for chain formation. So the polymerization will stop there. And because you have added small amount of dideoxy particular nucleotide in 4 different reaction mixtures, what will happen is that those will get incorporated at different lengths, right? So different fragments will be forming. And if you do some sort of electrophoresis or gel analysis, you will get this type of data. Okay. So from here you can see sequentially which particular uh, nucleotide is present from the particular the data that we'll get in the gel. You can analyze this through some sort of fluorescence or autoradiography and so on. Right, right. So this will give you the entire sequence from the short fragments after you analyze those. But of course, after the next-gen sequencing methods have been um, discovered or invented, for example, in this case, Illumina NGS sequencing, we will not go through that particularly. But just know this, that after the next-gen sequencing methods have been invented or have been used, so the entire scope of the sequencing that changed very drastically, right? So the next-gen sequencing method, the cost for the sequencing as well as the time consumed for the sequencing, that got reduced remarkably after invention of this next-gen sequencing. So here we have one particular graph. So it says cost per raw megabase of DNA sequence, right? So we have this kind of graph for that, and there is something called Moore's law also that is representing this slope. So anybody knows what Moore's law is? Any idea? So Moore's law particularly states that the number of transistors in an integrated circuit that gets doubled every 2 years. Okay, and it is, you can think of it in a way that it is a sort of a metric that can let's say quantify the improvement that that a particular field is getting. For example, in this case, in the integrated circuits and transistors and so on. So the computing power is basically increasing every 2 years, and that's a sort of good uh, comparison of a progression in the field, for example, right? So in this case, if any sort of field follows the same pattern of the Moore's law, then that field is progressing very well. Uh, in this case, however, in this particular plot, you will see that the cost per raw megabase of DNA sequence after somewhere around 2007, 2008. So there is a very drastic change here. So there is a very drastic decrease per the raw megabase of DNA sequencing, the cost, okay? And this particular point where this change is occurring, that is when the next gen sequencing methods are getting invented, right? Uh, so you see that it had a very big impact on the field, and throughout after the after the next gen sequencing methods are getting invented and so on, you will see at certain time at certain points in the plot, for example, this 2015-16, this time point. So there are a valley, like there is a sharp decline again. For example, here there could be a sharp decline again. So these sort of sharp decline or sharp valleys that you see in the plot, those will correspond to improvements that are being made in the next gen sequencing processes, right? So ultimately, the next gen sequencing processes advanced the field a lot. Similarly, if you have the cost per human genome sequencing, you see earlier the at the start of 2001, 2002, very big number here, right? A lot of zeros are there, so very high cost. And now, if you talk about 2021, 22, from up to that point, I took this plot. You can basically get your genome sequenced for like $1,000 or around something like that, right? So very big difference there, a very big improvement there also. So that's a so that's why the human genome project earlier took a lot of time, right? Because the cost and the resources it took was enormous, right? The time consuming, the time that it took to end the process or to complete the process, that was also enormous. Nowadays, for next-gen sequencing, it's not that big of a deal. Yeah. So in NGS, if we talk about just it's a bioinformatic process that can analyze massive data sets in NGS technologies. So DNA or RNA fragments, reconstructing of genomes, the genomic variations, their impact on gene functions. So comparing and contrasting similarities, identities, evolutionary relationships, and so on. Transcriptomics data, if you talk about, or if you talk about epigenomics data, for example, DNA methylation, histone modifications, and so on. So pharmacogenomics, personalized medicine. So all of these kind of related areas are where the bioinformatics could be again applicable in. Okay. So I think today I will just wrap up the class. Okay. The concepts that are left kind of I'll talk about in the next class, starting in the next class start, and then we'll also cover what Professor Kromiha goes over in his second week's lecture as well in the next class. So yeah, I see a lot of people had joined. So just to let you know that, for example, one minute, yeah. So to let you know that there is going to be another TA session held by another PhD scholar that will, I guess, be tomorrow, right? So you should have already received the link for that. So if you find the timing convenient, you can join any of the TA session. You can join both of the TA session. And if you can't join in some week, also we'll give you the link for the recorded lectures and the slides as well, right? So that's one thing. Those will be shared through the email to you later. I think you should have received the email for the session links as of now. Okay. So yeah, I guess with that, I will wrap up the session today. Again, thank you all for joining the TA session. And again, I welcome you all to the overall course of bioinformatics algorithms and applications. Please try to go through Professor Kromiha's lectures very carefully. If you have any questions, you can ask those in the dedicated forum for the course. And if you have any technical related questions, or any technical assistance, or some course forum or course server related questions, again, the admins of the course, they will also try to help you. Yeah, so I guess today that's all from my side, and thank you very much again for joining.
Thank you so much, sir.
Yeah, thank you so much. Okay, thank you.